So in the interest of time, because we have a sponsored talk, we have a lightning talk, and then we have two speakers, and we actually want to have an active discussion, I'd like to get started. There we go. So we're talking about sample controls, and in this particular case, our two speakers are flow cytometrists and a researcher. So sample controls cross all of our technologies, but today we're going to focus a lot on flow. Before we get started, I want to acknowledge that I am not Eric. He is the one who organized this, and he is the one who gets all the credit. I just had to show up and make sure everything flows well. So our two speakers are Shannon and Michelle. I'll give you introductions a little later on them. And our session sponsor, who's going to speak first, is Varsha.
I want to thank you all for coming here, and we are quite proud to be one of the sponsors. Hope you can see this. It looks a little grainy. But anyways, so I'm with Claret Bio. It is an NGS company, so not much flow. We, in fact, deal with cell-free DNA, so it's the stuff that's outside the cell. But samples and controls are a universal problem. And choosing your standards and controls cannot be an afterthought in any kind of experimental setup. And the challenges are you have low concordance with your standards. The standards don't mimic real samples. And these standards are not applicable across orthogonal tests. So if you used a standard for NGS, can you use it for flow cytometry? What are the challenges there? For our sort of focus, we deal with a lot of bad quality samples, especially cores deal with highly degraded DNA and RNA. It could be from FFP, it could be ancient DNA samples, and so on. And we get a lot of molecules that are not of the best quality. So within NGS2, there is a need for a standard that closely represents real-world samples and a library prep that can actually capture the data. So our flagship method is called Seriously. It stands for Single Reaction, Single-Stranded Library. It's a simple workflow where you start with your DNA or RNA input, you denature your DNA into single strands, and then you go directly into ligation. If you're familiar with NGS workflows, usually there are 7 steps that are involved. In ours, there are only 4. Because of our unique workflow, we have certain features that makes us applicable across various input types. So the problem is, so as I mentioned before, we have cell-free DNA, which is our key input type, and we work well with this. Now, the unique feature of our workflow is that it captures a lot of the degraded DNA. However, when you go into the standards don't mimic this. With our library prep, we get much higher complexity and so on. But then when you look at the cell-free DNA profile versus what the standard looks like, here you're looking at the profile of the library that you generate and the standard library, you see that it doesn't capture a lot of the features. And I just want to leave with reference standards. It gives us this cautionary tale where you don't capture the features of fragmented samples. And I know that in the rest of this talk, we'll learn about similar challenges in flow cytometry and how this can be addressed. With that, I would like to close my talk because I know that we have a lot of talks going on and I want to keep it relevant. But if you do want to learn more about our products, do stop by our booth D209. And I'd love to talk to you more about NGS and learn more about flow cytometry from you guys. Thank you. Excellent.
Okay, so I'm not sure how many of you were here for the introduction yesterday, but we do not have posters at the ABRF, and so instead, which I presume has been more productive in the past, we're having lightning talks, and we are fortunate enough to have Margaret Ushman from the Mayo Clinic, and she's going to give a flash talk on lightning talk entitled Starting Cell Number Limits for the MaxPAR Direct Immunoprofiling Assay for Mass Cytometry. Let's see if we can pull up your talk. That one?
Yes.
Yes.
Hi, I'm Margaret. MDIPA is, where is it? MDIPA is an easy-to-use human marker kit for mass cytometry. The original MDIPA protocol requires 3 million cells. Many of our client samples have fewer cells, but they would still like to use this assay. We tested protocol modifications to see if this is feasible. This illustration shows our approach. In the original protocol, 3 million cells were added to a tube of lyophilized antibody pellets. In modified protocol 1, we reconstituted this antibody pellet with buffer. 1/3 of this buffer was added to 1 million cells, and 2/3 of the buffer was added to 2 million cells. This preserved the original cell-to-antibody ratio, which we define as 1x. In modified protocol 2, this was for extremely low cell numbers, under 100,000 cells. The same method was used to reconstitute the antibody pellet. We removed some wash and centrifugation steps to reduce the cell loss during staining. The cell number was kept constant, and a serial dilution was made for a 3-point titration because of concerns of overstaining the limited cell numbers. In flow cytometry, overstaining occurs when oversaturating amounts of antibody make the negative population spread into the positive population. This reduces resolution of the populations. We examine this by staining 100,000 or less cells with different amounts of antibody concentration. Representative data is shown. The top row shows CD4 versus CD8 T cells. The bottom row shows gamma delta T cell subpopulation. Population resolution increased as antibody concentration increased from 0.25x to 1x antibody concentration. 1x cell population had the clear separation. Overstaining was not an issue. We then looked at how well our modified protocols identified lineage populations as compared to the original MDIPA protocol. We examined cell types that vary in abundance in PBMCs. On the left, data in yellow shows the standard protocol with 3 million cells. The same populations at same rates were identified in 1 and 2 million cells. The right shows protocol 2, and it gave similar results. The MDIPA antibody pellet can be reconstituted in MaxPAR staining buffer and divided among cells for successful staining. The MDIPA assay can be adapted for less than ideal starting cell numbers. Thank you.
So I'm not going to be cheap on time, but does anyone have any questions for Margaret? Okay, maybe afterwards. Okay, so we're going to start off with Shannon from, where are we? Sorry. Okay, so Shannon is an associate scientist at the University of Miami Miller School of Medicine and has been a part of the flow cytometry shared resource at the Sylvester Comprehensive Cancer Center for the last 15 years. She has experience in both flow cytometry and mass cytometry and is responsible for developing and teaching the facility's current courses on flow cytometry, which include instrument theory, compensation, controls, and panel design. Do you know which, is that one yours?
That's fine.
Okay.
Nope, that's hers.
Oh, sorry.
This one's mine.
I'm sure she's better at a PC than me.
Yeah.
Beautiful.
Okay. So thank you all for giving me the opportunity to do this today. And I wanted to ask first, how many people here have experience with flow cytometry? I'm just trying to get an idea of how many people. Okay. So everybody, good.
No basics.
Because I kind of changed my talk because I knew I would be talking to people who actually knew about flow and understand how difficult it is to get our users to bring proper controls whenever we do our experiments. So this is all part of a talk, or the teaching that I do already at the university. So what I decided to do today, since you guys already know this, is just kind of talk about what we do at the university, what we tell our users, and then at the end of the talk, after we talk to Michelle does her talk, we can have a discussion about what you all think. Are we missing anything? Do you suggest other controls? Because we know we don't see everything that's out there. You know, what might be missing. And then also, if you have any strategies that you've developed that have gotten it into your users' heads that they must use their controls. One of the biggest things that we deal with is that they just don't bring them. They don't want to. So we've basically developed this course, and how we teach basically is we talk about what are the most important kind of controls that you need to bring at a minimum when you're doing an experiment. And basically, we go through all of these right here as I do the teaching. So we talk about the unstained controls and what they're needed for. So mainly the control for autofluorescence. And when we go through our training, we actually tell them if they haven't ever used that tissue before, to bring them into the lab, and we'll put them on the instrument they want to use to look at the levels of autofluorescence and see what channels should we avoid. And as we build a panel, we know where to put different fluorochromes and markers and such. I know the second two are kind of controversial. Nobody really does this anymore, to do the starting voltages and also to set negative gates. But I do talk about it because you can still kind of do it with your unstained control, and I tell them that it's not going to be the actual way you do it, but it's nice to get a good starting point. And then we give them some instructions about what their control their unstained should look like. And I'm sorry, I thought I took off all of this coming in. So it's surprising to me, but we do still have to explain to people that their unstained controls have to be negative. It happens a lot. And they just don't seem like, people come in all the time and they say, oh yeah, here's my single color controls. And then we find out after we run them that they have GFP in them, or M-cherry or something. So we have put this into the training, and I stress this very highly, that they have to be negative. They can't have any endogenous fluorescence besides not having your actual markers. And then we do talk about, for compensation, the idea of cells versus beads. And we go a little into, if you're going to do that your source of your unstained control needs to be the same as the source of your single color controls because of autofluorescence needing to match. And basically, we tell people, if you're going to mix them, to come talk to us before you do it, because then we can talk to you about the best way to do this. And actually, a little bit later, you're going to hear from Michelle, some really, really nice research that she did recently, and which is things that we have been telling our users for years. But now it's nice because we have a nice paper that we can show them when they're going to use cells and beads together. The biggest thing that we can't always get through to them is that your cells have to go through, if you're going to use them for controls, have to go through the exact same treatment as whatever you're doing to your cells, because they change the properties. So we have issues all the time where people bring in and they've stimulated them or they've treated them with drugs and they don't have the right controls. So we go through the training and try to get to them and say, listen, if you're going to treat them with a drug, you need to bring us a control with them treated and non-treated. You have to have basically an unstained control for every single element of either stimulation or drugs. So that's the first part with the unstains. That's sometimes a task all by itself. Then we move into the single color controls. And we explain to them why you need them because, for newbies, they don't really understand and talk about compensation. So this is part of a whole, my first set of classes, which I talk about the theory, I talk about compensation, and I talk about single color controls. So by this time, I would have already talked about compensation and spillover and everything. So we explained that this is what you need those single controls for, is for compensation, and that you must do compensation. And why you need the single color controls is because you can see the spillover, and then use it to start your compensation process to get to your true signal. And then we start to talk to them about, sorry, I think I pressed the wrong button. Where is it? What we think is the best practices for single color controls. One of the things is they have to be positive for the marker of interest. And I don't know if anybody else has run into this, but a lot of times people bring in isotype controls and say, this is my single color control. And we have to explain, no, that's not going to work. We also have issues where people want to bring GFP for FITC, or FITC for GFP. That happens a lot. Usually it's FITC for GFP. And while technically, since it reads in the same channel, in conventional cytometry, we could get it to work. We have done it in a pinch, but I explained to them that they're not the same. And so, especially in spectral, you just can't do it. And that if you're going to have this fluorochrome, especially the endogenous one, you have to find a control that has GFP in it, or M-cherry. And then you have to use FITC and not use Alex448. Yes, they read in the same channel, but they are not spectrally the same. And then we also talk about tandem dyes and all of the problems that tandem dyes have, but in particular that if you're going to use a tandem dye, with your single color controls, it has to match what you're using in your experiment. So you can't take the last of one vial for your single color controls because you have just enough for that, and then use a totally different lot number with your samples. And in this part, I actually go into tandems and what they are and how they work for people who don't, for the newbies basically who don't know. And all the different problems you have with tandems and how they're temperature sensitive and light sensitive and they tend to fall apart. So they have a good understanding of why, we're particular about this, especially since, again, they can vary from vendor to vendor, from lot to lot. There's all kinds of potential issues. But one of the things that we have started doing as a way to teach them about good controls is also understanding what they have and making sure they know that they have tandem dyes. And this is one of the slides that I give them because a lot of people don't understand, when you have PECY7, we know that it's kind of in the name. But if you have the brilliant violet dyes or the brilliant blue dyes, people don't understand that most of those are tandems. And so you have to treat them like tandems. So this is one of the slides that I put in just recently, just to kind of show everybody that a lot of these dyes out there are actually now tandems, and so you have to be careful with them when you use them in your experiment and as controls. And then this is actually something, the next slide, that I would like to discuss in the discussion part, is how many people use the special buffers for these tandems. I've heard some people use them. We recommend to everyone that you use them, because of the data that we've seen from the manufacturers. We do want to test this eventually in our in-house, just so we can show, I feel it's more constructive when you show people like, this was done on this aurora, and look, if you don't use these buffers, this is what happens, and when you use them. But I'm really interested to hear from everybody else if you include these as one of, it's not a control, but it is kind of, because if you don't have it, is it really affecting the dyes? Are you seeing that? Are you not seeing that? But we do teach them that these special buffers exist and that you should be using them. So overall, though, for single color controls, we say that we think that it's the best to use your cells for your single color controls whenever you can. That's going to be the best representation because you're going to match your autofluorescence, spectral properties, and everything else. You can use we tell them we can use the antibody capture beads if you have to, so you don't have no idea what your expression is going to be like. You don't have a control that you can get a hold of, or your cells are just limiting. And then again, we talk about the fact that you can use them together as single color controls, but then we also talk about what you have to do. So you can't use it universal unstained, and how to do that when you're setting up compensation. And then we just reiterate, it's got to be your antibody fluorochrome combination. So if you're using CD25 FITC, your color control should be CD2550. And then we see this a lot, and I don't know if everybody else sees this still, and I know there's controversy about this. We tell people not to, if they can avoid it, take something like CD4 and use it as a compensation control for something that's a lower expression. Now, I know some people do that all the time. I know people who use just all CD4 beads or CD4 antibodies for their single color controls. And again, that's something that I think would be interesting to talk to in the discussion, because we suggest they don't do it, but I know a lot of people do it. So we actually suggest it's maybe better to use compensation beads, but it may not be. But what we do tell everybody is that no matter what you use, if you do that, because there are some PIs who absolutely want you to do that, and we understand, like with some of our graduate students, even undergraduate students, that they're going to have to do what the PI tells them to do, that you have to make sure that whatever you do use in that case is going to be at least as bright or brighter than the intensity of your actual fluorochrome, or else your compensation is not going to work. And this is really, when we talk about single color controls, the thing that we hammer home a lot, because what we're finding is that, especially with the Aurora, we have an Aurora with the spectral cytometry, is there are a lot of markers that are not high enough on beads. They're much higher on cells, which is why we usually say we think the gold standard is to always use cells if you can. And that actually has been hard for some people to do. They don't seem to understand, and we show them. They come in with the beads, and then everything's is not compensated properly because their cells are so much higher, like CD45, Ly6C, Ly6G. There's a bunch of them that we all know are super bright. So this is something that we try to really hammer home. Unfortunately, sometimes it takes them actually seeing it on the cytometer, and by that, sometimes it's too late. But we do hammer that home. So that's what we do for single color controls. The next one that we do is viability controls. I'm sure a lot of you have trouble getting people using them. For some reason, where we are, people just don't want to use them. So in the training, I try to explain to them what dead cells do. They have an increased autofluorescence. They take up antibody non-specifically. And it's going to lead you to, false interpretation of your data. And now you've spent 6 months on a project and realize that these are all dead cells. And I tell them during training, I'm like, please don't do that. And I like to show them this picture. Just when you take the dead cells out, you clean up these populations. And while, the one on the left, I would never assume that was real, but there's that one population on the right-hand side that might be real if you don't know what you're looking at. So we really try to talk about using a viability control, and that there are so many of them now that there's no reason not to do it, no reason to not have one. And then we just do a discussion of all the different ones that you can have. So again, there's no reason not to have one. You can either use one of the DNA binding ones, but if you want it to be fixable, then there are a ton of the protein binding dyes. And so I actually give them a list of like what you can use for the DNA binding ones, and then there's another slide after this that has all of the companies that make these viability dyes, so go look and find one. And we also stress that the dyes are fixable, the protein binding ones, because then you don't have an excuse of, oh, I'm doing this big huge experiment and I don't want to have to put a viability dye and then run them right away. Well, you can get a dye that's fixable, and then you can come the next day and run your samples. So we have FMO controls, which I don't know how many people, everybody should be using them, at least with the Aurora at this point, in these high parameter panels. These are also ones that we have a lot of resistance to people using. They don't understand why, and when they're doing a 37 color panel, they don't also want to bring 37 FMOs. So the way we're training them, we're trying to explain to them and show them that it controls for the spillover spread. Your compensation is correct, but you still have spread, and you need them to do your population gating. So we try to say, okay, do it the first time, see what you really need. Maybe you only need 5 of them. You don't need 37. And some people do it and some people don't. But then we just go through an explanation of what they are. And I like to show a figure like this to really demonstrate that you can, again, misinterpret your data if you do not include an FMO control for these, especially in the high parameter panels. But this is a 3-color panel. I know this is a really old figure, but I think it shows really nicely that if you don't have these FMO controls, you're going to assume that you have a positive population that just doesn't exist. So we stress this one a lot. So next, isotype controls. I'd love to have a discussion about isotype controls. I don't recommend that people use them because we all know that they've been generated supposedly against things that are not in our cells, but we know that's not true in a lot of cases. I also know that some papers still require them, some PIs still require them, or some reviewers like to see them. So we kind of talk about what the limitations are and that you should try to find one that's matched already by the vendor who will try to match it for low nonspecific binding. And I like to show them this picture to show that this is nothing, it has nothing to do with your actual specific antibody. This is just nonspecific binding of this isotype. But what I have learned recently, another way to teach it is that they can be used to figure out whether or not you need to do FC blocking. So then I kind of move into, well, what are these FC receptors? Because a lot of people don't block. And some of them don't understand, they don't have the immunology teaching to understand just how nonspecific binding occurs. So we go through a whole discussion about what FCA receptors are and how the cells are actually binding to the constant region of the antibody because they have these receptors, and so you're seeing this signal, but it's not real because it's not actually binding to the specific part of the antibody. And so I talk about how, FCA receptors are on so many different types of immune cells, and that if you have a specific, depending on what sample you have, you may or may not need these receptors. So you have to know what you're using, what you're looking at to know whether or not you need to use FC block. And that if you see something that's weird and you haven't blocked, that maybe you need to go back and do FC block. And then again, I like to show them a picture of what that looks like. And again, a one where you might think that that's a real population if you backdating on the monocytes, but when you block the FC receptors, you see, nope, that's not real. So you don't spend months. And then I always like to give them options about how to block. So either the normal serum, and I give them a list of different FC block they can get from a couple of different vendors. And then finally, we'll talk about biological controls with them. So those are ones that we know whether they're positive or negative, and the positive is great for when you don't know whether your cell type is going to express. So you have the positive control to make sure that the antibody's working and you can see what it should look like, and conversely, the negative control. So it's not supposed to express it. So if you see some binding, then you know that you have some issues where you have to do, there's a lot of nonspecific binding, you have to maybe use FC block or do some other optimization of your protocol. So finally, this is a very shortened version. There's a lot of extra slides that are in there. We go pretty in depth. But this is the question. How do you get your researchers to use the appropriate controls, especially considering how many there are? And so what we found is working is that we've created these in-depth classes for our new users, whether they be technicians, graduate students, postdocs, PIs, that haven't done flow, and we really talk about all of these concepts in depth. And one of the other things that we've started doing is that we now have, a proficiency testing for people who want to use self-use. And in order to do that self-use where it's 24/7, 365, they have to go through my training. So there's basically 4 hours of theory between those, instrument theory, controls, compensation, and panel design. And then we also do some training on actual instrument, how to use DIVA. Most of our instruments are BD instruments. And then after that, you go for hands-on training. And in particular for one of the ways we've been dealing with this at the front side is that we, for the Aurora, since spectral is fairly new to us, we have one-on-one trainings. And we sit and we talk about all of these things, about all the controls you need to bring, about the titrations that you have to do, all of those elements before they even start designing their panels. So they're not trying to go back and optimize panels. They're doing it from the very beginning. And we also want to work on generating some real examples in the facility. So this was done on the Aurora. This is what can happen with your data. But what I have noticed is that since we started implementing these trainings a couple years ago, is that we are having less problems. I have graduate students in particular come in and start a new project, and they bring their controls. And it's been good. They bring all of their controls, or maybe they forget one or two, but it's so much better than it used to be. We still have people that just won't do it, or forget, or don't want to do it. But that's pretty much, this in-depth education from the very beginning, before they even start, has been what's helping us the best. And then unfortunately, the second best is them coming with all of their samples and realizing that they can't do their experiment because they don't have their controls. So I guess the reality of the situation has been the second best teacher. But yeah, it's still a struggle. I hope one day it won't be, but I don't have high hopes about that. So thank you. We're going to leave questions until after, and I wish I had a picture. I have a picture of our facility, the staff. There's 5 of us at UM. One of them sitting right here, our lab manager, Patricia, and then we have 3 other people within the facility that we all are struggling with the same thing. So that's it. Thank you very much.
Thank you, Michelle. So, Shannon. Michelle's the next one. So Michelle is an assistant professor at East Carolina University in the Department of Microbiology and Immunology. She is interested in human immunity, specifically focusing on age-associated changes in the hematopoietic progenitor pool. And will you be able to find your slide? If not, he'll help you.
Oh, no, not that
That's
one.
not it.
I think it was, there it is.
There it is. Beautiful.
All right. I want to first say thank you for inviting me to talk today. So I am hopefully not one of the more irritating people that you have to deal with, but I am on the side of the irritating people. So I'm approaching this from the standpoint of what I, as a PI, would like to have happen when we're doing flow cytometry experiments. So we've progressed a lot since flow cytometry started becoming a thing. From when I actually started working on flow cytometry, I worked on a 1 laser fax caliber on FCS1s back in the day. And so from the last time I checked the Cytech website, we currently have 360 different fluorochromes that can, fluorescent proteins that can be used for flow cytometry. And the most stains that I've seen so far published is a 43 color panel on human PBMCs, which makes me happy because I look at human immune cells. So I saw that and I'm like, we can stain for all of the things. And so a lot of what I'm going to be talking about is done on the spectral cytometer, but we did do it on a conventional cytometer as well, so it applies to both of them. So here's where the problem comes in from my side. We see in both of them, there's going to be peaks popping up in different channels. So just a 35 fluorochrome and fluorescent protein panel looks very, very messy. And so my response after seeing that was, well, okay, so we can't stain for all of the things. So, oh, that was not supposed to be there yet. So large panels are messy and complicated. So how do we properly control for those large parameter panels? All of the commercially available beads effectively work for all kinds of large panels. It's what we would assume. As researchers, we assume if it's on the market, then it works just the same. All of them work the same, and they work for everything all together. So do they actually control and compensate effectively amongst all of them with cells? And then how often do we need to run controls? At the beginning of the study, once a month, once a week, every day. I know somebody who actually ran a control at the beginning of the study and spent months running everything off of the same controls, controlled everything off of the same set of controls at the beginning. I also know somebody that made a giant tube of every single control and just kept running the same one over and over for months. I didn't do that. I was using mice. I had plenty of cells to do my controls every time. So when I moved to East Carolina University, we moved to a spectral cytometer. I'd never used one before. And then we started running some of my panels, and there was weirdness in those data. Didn't look the same as I would have expected from my previous studies. So from that side, and then my collaborator, who really just wanted to see what side-by-side of the various beads looked like, we wrote a paper, did the experiments and wrote a paper, which actually for him, said, this is what you need, what we see from these beads. For me, said, what do I need to do to control my samples so we only have to do it once? So to start that, we looked at the actual antibody binding sites on the beads and how they are actually different between the different fluorochrome labeled antibodies. And the green means it's pretty close to what we see on the cells. And so this is CD4 to CD4 on the beads to CD4 on the cells. Yellow is acceptable, and the white are not really in the range of what we would want them to be. So NovaFluor, for example, is not good on anything. I personally don't like the NovaFluors. I don't think they're good long-term at all, but at least with the beads, they don't work very well. So the data that I'm going to be showing is based on what we called a median mismatch index, which is basically you look at the MFI of your negative population and the MFI of your positive population and use those numbers along with the negative RSD to come up with how close of a match you have for the straight line from your negative to your positive. So the closer it is to a straight line across, the lower the number, the better your MMI. So just to give you an idea of what this looks like. Does this go on there? No. Okay. In the first column is the green numbers, so those are pretty darn close to fairly straight with the negative and the positive. You start to see a little going up and down for the second column, and then the third and fourth column are just not going to be very good at telling you if it's positive or negative or not. Some of them are really bad. Some of them, like the third on the top, there's a ton of spread there that's also causing problems. So this next panel, or this next slide, it's very small, I understand that, but the point that we're getting from this is the green, the red, and the yellow. So the top row that looks mostly green, that is cells compensating cells. The rest of them are cells compensated by various commercially available beads. They're not great. And so this is doing single colors. So this is PBMCs stained with CD4 compensated by beads stained with CD4. So this one here is cells compensated by the beads. Now you would expect that the beads should be able to compensate themselves as well. And sort of. So this is each repeat of the bead samples are compensated by their own cell or their own data. Some of them are pretty good. Some of them are terrible. One of the prevailing things that we noticed was that the violet is often not great. So the violet channel has some issues with, especially on the spectral cytometer, with compensating with beads because they have their own autofluorescence in the violet channel. Okay, so that answers the first question. Are all commercially available beads equally effective in these kinds of large panels? And no. So then what this tells me is, depending on what you need, you're going to need to test your fluorochromes in your panels to comp beads. And if you have to use beads, you're going to need to mix and match to fit what it is that you're looking for. You can't, most of the time, you can't just pick one bead set and have it work, which is where I had the problem because I was using UltraComp, and that one's not great. Okay, so then the second question, how often do we need to run the controls? Everybody in here is going to say no, that it's every day, every time you run your samples. But how do you make us realize that? Because as a PI, I'm going to say running controls once a week or once a month is going to be way cheaper for me, but at the loss of the data. So if we look at running so this is looking at the peaks for FITSI and the PE Saigh 5, and it's going to be on the cells and on 5 different beads. And it's subtle, but they don't match up. And so the small changes equal not great changes in the math and the algorithms, and that's why we see all of those red. So then if we look at the same set of beads run multiple times, so in this case we used MAC beads, there's also slight changes in there. It doesn't appear to be that big of a difference in these peaks. However, it does translate into not great numbers in the actual algorithm output from these data. So then what happens when we look at cells? So cells are the ideal ones to use, but everybody's like, okay, I can make my controls out of cells and just use them over and over because they're the cells. Except if you run the same set of cells 3 days in a row, they don't match up. So over time, even with the same cells, there's going to be variation in those fluorochromes, which is going to cause problems with being able to use those data, at least from a control standpoint, to control for other days, other stain sets. And you can actually see that in the numbers. So this data is looking at day 1. So this is 3 separate stains. So one set of stains, the first stain, is going, in this case, the top is set 1, compensated or unmixed using set 2, and then the bottom set is set 1 unmixed by the 3rd repeat. Now this is all PBMCs. These are the same donor, so there's no difference that way. It's all the same donor. I have aliquots that we just took the same donor at 3 different thaws. And the mismatch is fairly even in what's happening. You see the same things are mismatching. But there is mismatch. So the controls need to be done the day that you're actually staining those samples. You can't use the same set over and over to compensate. So run new single controls every time you run. But how does that translate to an actual full panel? Because everything has been single colors on cells and beads at this point. So we devised a stain set, a small stain set, because we didn't want to do overwhelming amount of analysis on this. But we used DAPI as our live dead, and then we looked for CD3, CD4, CD8, CD19, and CD45RA. Very simple panel. So if we look at just the traditional user-driven analysis, we can see changes in multiple panels that we have looking to one by one. And with these beads, so the top is the cells, and then there's beads underneath. And the beads are showing different, they're showing different patterns than you would see from the cells. Some of them are fairly big differences. Let's see. Did I already? No. I guess I don't have it on here. But if you look at the third panel and the bottom set of those panels, so we've got a really nice, not really shifted looking population. But then if we look at the beads at the bottom, they're starting to shift up. So you're getting stuff that is not positive shifting into the positive, and somebody that doesn't know that that's not really a low population may think that that's low expressing when it's really not. But then so now people are doing more unsupervised, like t-SNE plots and UMAPs. If you look at the t-SNE analyses of just that data, the top being cells, and then the bottom being 4 different beads that compensated those data, we can see a lot of differences between what the cells do and what the beads are doing to those data. So that's going to make it more difficult to be able to have somebody look at what you've done and try to repeat it and say, well, that doesn't look the same. So from the PI side of it, that means I can't prove that my stuff is reproducible if nobody else can do it. And with the NIH saying we need to hammer on reproducibility, that's going to be a big problem. So one of the other things the NIH is wanting us to do is share our data, so on repositories. For flow right now, that means the flow repository. Some people are doing it, some people are not yet. They're eventually going to have to share all of that data. So if we look at the data, these are from OMIPs, 4 different OMIPs. All of them were compensated using BD beads, which from the data we have are the best of the options that are on the market right now. And we still see there's some compensation issues. So there's mismatch, median mismatch index problems going on there, which means there may be some cells that are getting analyzed as positive or negative that shouldn't be, which again is going to be a problem for reproducibility and is going to affect your users' reputations. That's what you need to do to get our attention, is tell us it's going to be a problem for us reputation-wise when people can't reproduce the data. From the student side, doing all these controls means it's going to be easier to analyze the data. Okay, so how do we properly control these large parameters? It's best to use cells if you can. Sometimes you can't. If you're doing small volume human studies with PBMCs and you get, I don't know, 5 million cells, it's going to make it very difficult to do large panels to maximize the number of your cells using cells for your single color controls. You also need to design the panels based on the marker considerations when choosing the fluorophores. Some fluorophores are just not going to work on some cells in certain colors. And so that's a problem that us as researchers have. We look to see what's available, not what will work. And then you have a bunch of antibodies in your fridge that you can't do anything with because they just don't work for what you wanted them to do. And then if compensation beads are necessary, we need to look at the antibody fluorochrome compensation beads themselves, because there's going to be variation in how well certain beads work with certain fluorochromes. And if we don't test those, we don't know which ones we can use. So that's the end of my part of this. I wanted to thank, so Sarah Lowe is my graduate student. She did some of the work. Debajit Braumik is our core director, and he did a lot of the work on this. So they were very helpful in me being able to say, hey, look what we did. And then my part was largely funding it. That's important. So the QR code is for the paper, and I guess it's question time, right?
Yeah. Thank you. Yes.
I would just like to commend you don't want to diminish what you did here today because it was super, super important. I've been running core for almost 27 years, and I've never had a PI come to me and say, I'm going to I want my staff to run every control every time, and I want to do all this in-depth testing. So thank you.
Oh, my student.
I'm sure you were preaching to the choir.
My student went even further than that. Every time we get a new antibody in, she does the titers and then she does the full stain with it in triplicate to make sure it actually is staining correctly.
But what I would also implore you to do is whatever meetings you go to, can you present that in one of your workshops? Your fellow PIs, especially in the not so traditional flow cytometry group, meaning,
It's the people I work with, really.
the cancer biologists and the molecular genomics people who are diving into TIL work, because, they're not immunologists, but they play one on TV kind of thing, and they want to do these huge panels, and they have no idea what they're doing. That's the kind of stuff they need to see as a starting point for them, because it's a realistic, it's real work that needs to go into this. It's not magic. Howard Shapiro is listening. There is in flow cytometry, it's zero law, right?
So for our core facility, our director basically doesn't let anybody touch the machine until he's looked at their experiment, helped them with the panels, helped them with titering the antibodies, and gotten everything set up with the controls and everything to make sure that they don't mess up our machine, really.
Yeah, I mean, the bigger the institution, the harder it is to control every user that walks in.
Well, that's where we're a small institution, so we have the Aurora and we have a sorter, and sometimes the Aurora is fully booked. Hardly anybody does any sorts, so it's picking up. But right now, it's more on the slow side. So it's a lot easier for him to look into some of these things while still making sure everybody is doing things properly.
So this session is being recorded, and you can probably make this available to your users or maybe implement it as part of your training.
That's actually what we're probably going to do is, so Michelle and I actually, she went to the same graduate school, so I know her for a long time. And she used the facility when I was first started working there. So she called me about this paper and she showed me before they printed it, and I was like, this is amazing, and yes, I'm going, once you publish this is going to become part of the training. So, because this is stuff that we've talked about amongst ourselves and the staff. We know everybody needs to bring their compensation controls, but stuff like this is awesome because then we can show our users and say, look, this is why we tell you. Yeah. Exactly. So I plan, this is gonna
But
go.
yes, from
yeah.
from my side of it, having to repeat things because we didn't do the controls right is a pain.
Yeah.
It's
And
it's expensive, and I've also been the stupid graduate student that did it wrong. So I've got both sides of it now, and sometimes it takes us a little longer than others to bang in our heads to do things the correct way. But it is helpful to start with, you need to do it the right way so you don't have to do it 600 times.
Yes. Right here.
Yes, sir. So we discussed this paper with a few colleagues, and we come to a point where in discussion where we thought that we might be coming into a place where there's not going to be perfect controls for some experiments, very large panel, especially when you start playing with cancer cells, and now the rules of like matching your autofluorescence becomes a very complex affair. My question is, at some point, what do Better. So what do we do with like any kind of spectral problems that might be still occurring?
Okay, so
When do you apply a composition on top of your mixing?
before you answer that, I want you to restate the question, because I just realized again that we're recording it, and I'm going to walk around with the mic for the rest of the questions.
So the question basically is, what do you do when someone has all of the good controls and it's still not unmixing properly? And I do have an answer to that. I don't know if everybody would like that, but that's where mass cytometry is really, really good. Because of, no? Why don't you agree? I'm just curious. Okay. I gotta get my glasses.
So first off, you're telling a bunch of users that prepare to experiment on spectral flow, switch platforms and go to mass cytometry. It's just not
Well, we try to tell them in the beginning,
You-
that's
you
why
don't-
they come to
no, but
us.
the point is, you won't know until you get to that point where the controls are just.
So that's one reason why we ask them to come in the very
Right.
beginning and look at their stuff on the Aurora or whatever they're trying to use to see what kind of autofluorescence it is. If there's any clue that we might have that doing flow is not going to be the right way to go, that maybe you need to look at something else before they get into it. Now, I understand, you've already got,
I understand your point.
Yeah.
I'm just saying it's just not the response I'm going to give to my users. So you need to like, mass cytometry is substantially more expensive, time-consuming
Yes.
affair. It's not something you can tell them. So again, we just like the solutions we've been telling our users, like go back, make better controls. At what point do we say it's going to be okay to publish data that doesn't look properly compensated, or do we need to correct for that to prevent reviewers to fidget about the way the data is presented?
So this is going to go back to where I was saying that if you have to use beads, you need to test what they're going to look like. So some of the beads work great on certain colors. And so unless you're trying to do a massive, massive panel, it's easier to say these beads are going to compensate or unmix with an unstain that you can subtract the autofluorescence with sufficiently, and then you can do maybe a little bit of tweaking on a compensation panel to fix some of the stuff that the algorithm isn't picking up. If you restrict yourself to these fluorochromes that aren't going to be terrible for that environment, like I wouldn't use beads on violet. In my experience, the violet laser doesn't work well with the beads because there's a fair amount of autofluorescence in that channel. But they work reasonably well for not the fitzy part, but the blue, yellow, blue, and the blue laser, the green, and red laser. They tend to unmix better for certain bead sets. So that's where figure out for the colors that you want, for the markers that you want, that you know you can get in those specific colors, which beads are going to have the least amount of problem for your panel. That's what I've had to do, because there are some things that I can't use PBMCs to stain for. Anything that's going to be in the hematopoietic progenitor pool mostly are not going to have enough in the mature population to be able to compensate, so we have to use beads. So I had to find which beads work for those colors.
I've seen some data published that does not look like it's compensated perfectly for whatever reason. And personally, I think that if you can show that what you have is still real, valid, that it's okay. My concern comes when you get into the high parameter analysis where it's unsupervised, where the algorithm might say that this is positive when it's not. Which of course you can always go back and once you have those clusters, go break it back down into dot plots and do some other analysis with it. So I guess if you can show that even though you can't get the compensation perfect, but you can still validate what you've done, then I think that would be okay to publish. I mean, as long as, again, you show in your paper all the steps that you took And that, you know, I think that especially as we get into more and more of spectral, and now, of course, we're trying to find more fluorochromes to fit in those little spaces that don't have things, it's going to become a bigger problem with the unmixing, where some of it's going to be pretty heinous. But if you can prove that it's real, then I would believe it, I guess. As long as you can, again, show me all the steps that show that you've done to show that, yeah, this looks not that great, but it is a real result. And that it's unavoidable with the panel. I think that would work.
So also to be fair, most of the people that are going to be reviewing those papers don't know enough. So
But that scares me.
I know, but if it's, in your opinion, looking at it as the expert for this cytometry, looks reasonable enough to say, yes, that is real, that's a good population, it'll pass muster for us, because we'll read it and be like, yeah, that looks good.
Yeah, thank you so much, Shannon and Michelle. And I was literally I was so tired when I got in, and when I saw all this presentation, I was like, yes, preach. Thank you. I totally agree with what you guys were saying, but to answer or add to what you were saying, I would go with pick the best, don't just go with all or nothing. I can't just do cells, I can't just, it can't just be beads or cells, right? You could actually pick the ones that is going to be better on cells, pick the ones that are better on beads. So I used to be in a core. So when I was doing my training, I usually tell my people to bring a set with cells, bring a set with beads, and let's see which one is better at mixing. And we'll pick the ones that is doing good for the fluorochrome combination. So that's how I addressed it when I was training my people in the Aurora or the high parameter flow panels. So that's what I would do. So thank you so very much. I appreciate that. I did have a question. Yeah. Going back to training your people on their samples, right? Do you prefer training your users on their own samples, or do you have a training set?
We don't have a training set. We prefer that they come in and bring their actual samples, because for a variety of reasons. One, we don't have what we need to cover everything, because we, as part of the Cancer Center, we service everybody in the Cancer Center and then also the rest of the university, and there's just too many different samples for us to really have something effective. And then it's real to them, because it's their samples that they've spent time working on it. It's just more like, we think that just, it sinks in more.
That's true.
When you
that's
teach
true.
them with their samples. So we'd like to have them bring their actual samples in.
Yeah. So I came from Baylor College of Medicine, so I worked with Joel Cederstrom in his core, and we usually just trained with our own training kit. We had our 2-color, 3-color panel, and for me, I was doing the high parameter unit, so I was training with my own PBMC panel. So I was just wondering, really just curious about trying to see whether you have a standard panel or whatever they brought in.
So our facility actually does a combination of the two. There's a set of things that Debajit will run through for staining, and it's things that gives them the basics of how you do the staining. So he'll do a basic live dead thing where he physically shows them, for control for the live dead, how you set up for those controls with the heat killing, and checking. This is where some of it will be their own samples, checking to see if heat killing is sufficient or if you have to do something else, because the heat, it either is all or nothing with the heat killing.
10 seconds, 8 seconds, what do you want to do?
Right, so, but then he also goes through cell cycle staining, because that will give them an idea of how different intracellular stuff works, and then he'll do one other training that I can't remember right now, and then so that's the first day. The second day is staining their own samples.
Gotcha.
So it's to get them, I guess, comfortable with staining things they're fairly easy stains, but something to give them a basis for, hey, you got this right, now you can get your own right.
Thank you so much. I appreciate that.
I have kind of a follow-up to the training question. So that answer implies you do that kind of 4 hours of intensive training with a bunch of theory with each user individually when they come to the core? How
No.
do you structure it so you get to catch everybody? Like, for our imaging core, we offer a course once a year, but then that doesn't always catch folks on the cycle when they're coming into the core.
So we actually do training every 2 to 3 months. We do it virtually through Zoom, and I also record them, and so I make those recordings available, and they have the PowerPoints available, because I put a ton of links in my PowerPoints, because I like them to, look up the links for the different products and the papers I have in there and such. So we'll do it more often, actually, if we get enough people that come in and say, hey, we want to do it. So it's done on a regular basis.
Do you think people watch the recordings?
I do have people who watch the recordings, yeah, I do. They have to take a proficiency test. So in order to get that 24/7, they have to pass the test. So a lot of them do actually watch it, because we throw a lot of information at them. And that's another reason why I gave it to them. And I have to say, it is actually, I think, really working, especially for people who are new to flow or the new graduate students. They come and they say, oh yeah, you're telling me this, and so I did this. And it really makes me, it gives me hope that maybe if we keep going, because it's really the old timers like us. Like I started and there were like 8 fluorochromes, and now we have way more than that. So we really focus in our lab on trying to train the heck out of people. Like Patty does a lot of Patty came up with the training for the Aurora, and when we got the Aurora, she sits down with them before they're allowed to touch the instrument, and goes through all of the little tips and tricks and quirks and things, and how to do the panel design and things that she's noticed, and so they get a lot of training from that at the very beginning for spectral. So.
The one-on-one hands-on training can vary anywhere between 4 to 8 hours.
Yeah.
It depends on their level of understanding and the instrument. And we have regularly 40 color panels on the Aurora.
Yeah. They don't always look good, but we're working on that.
Well, I have a 36 color that
Yeah.
is pretty good.
Yeah.
So I have a question for the group. Are there any solutions that you have stumbled upon in your course to help your researchers actually run the proper controls? I'm sure everyone has struggled with it, but maybe some people have found a solution that they could share with the group.
No? Nobody? Nobody? That makes me
So
sad.
how many researchers come to you guys when they've designed the experiment, but before they run it? Because, that would be a solution.
They do sometimes, yeah.
Just not often enough, obviously.
It depends on how much experience they have. So if they feel like, I've done flow before multiple times, they're less likely to come. The spectral flow, I do have them come more often, and I do have an SOP that basically takes them through how to design a panel.
Mm-hmm.
And that seems to help a lot. And then I tell them to send me their panel before they purchase any antibodies, and most of them will.
Yeah.
Send me the panel.
We do the same.
And just to try to catch a couple, and I'll say, these two things really, the difference is 0.97 and 0.9, maybe that's not a good one to put
Yeah.
together in the same.
But I think we have a problem with researchers who know old flow
Yes.
and teach their staff, graduate students. We have a big problem where they're using a viability diet and then just using, in their case, this is the Fixable 780, they're just using CD4. And then when it doesn't look good, they're like, fix it.
Yeah, we get a lot of that.
Sort it. So I have to make decisions at that moment. And I have a lot of problems with red. They're like, oh, well, my cells are medium. You don't need red. Get the medium, get red. They still don't know, because they don't know what red does. So those are the biggest challenges right now.
Or you get graduate students that they know what they're supposed to do, but their PI says, no, don't do it that way.
Or I have too many controls. I only have
Yeah.
3 samples, but I have to have 30 controls. Yes, you have to have 30.
Yes, you have to have 30 controls. And then when it doesn't work, because they didn't bring the controls. They're like, why doesn't this work?
One of my favorite is put up the unstain, there's GFP or and
It happens way
there's
too often.
dye, and they're like, well, I don't care about it, but it's still in the sample.
Yes, that's what I keep telling them. It's like, it's still there. It's still causing problems.
Well, I did have the person that kept putting a live dead stain in every single sample.
We have that too, yeah.
They did it so many times, I told them I was gonna, if they did it again, I was coming to their lab and throwing it out.
That's when you put it in a tube and tell them that they can check it out from you when they get to the flow lab. Yeah. You just put it in a box that they have to smash the glass. So first of all, I think we need to make an online button that we can all just go hit when somebody comes in with GFP positive cells for all of their single stain controls. Because I want to see how many minutes it takes for that to hit a 4-digit number. But I wanted to share a couple things that we've done in our core. First of all, I have a giant box in the refrigerator called capture beads, and I have as many different kinds of antibody capture beads in there as I can get, so that if we've got somebody who says, well, this is the beads that I had in my lab, so that's what I'm going to use, that when they, especially if it's something so egregious, like I appreciate you did so much work, your people did so much work putting it together. That was example, the matrix of all the possible ways you can do the compensation. Sometimes there are things so egregious that you can see them while you sit at the instrument. And that we've at least been able to kind of stem the hemorrhaging by saying, here's the box of beads. I think you should try this one next. And our goal isn't to supply the beads for the entire university, but if somebody in the moment is like, here, this is what, this doesn't look right, help, that I have the beads there.
That was the reason that I started saying when I switched to the Aurora, I need to find a different bead set because the beads that I had looked horrendous on the spectral cytometer. And I was like, I can't do anything with this. I can't tell what I'm looking at.
And the other thing I wanted to mention about the idea that especially when the grad students have the best intentions and the PIs say, no, you can't run that many controls for this experiment that only has 3 full stains. We have a staff repository called email templates for common responses so that we don't have to keep recreating the wheel every time. And one of them legit is a 28-point font Word file that says, it is better to have and not need than to need and not have. It's like, just to speak to your point about the first time you run it, just bring all the FMOs.
Mm-hmm.
And then you can figure out which 3/4 of them you can drop for the next one.
Yeah.
But what do you do when you get there and you don't have it?
Mm-hmm.
So,
It's frustrating.
I might increase the font size on that one, but it's, yeah.
We should make a big neon sign, put it in front of the door.
One more comment.
It's actually like, do you guys recommend any internal experimental controls, like running a rainbow bead, so that they can actually have something continuous, like if they come back in a year and say, hey, I want to run this thing again, so you could.
I've started talking to people about that because now that we're getting again into the high parameter stuff where we're doing concatenating everything, I'm starting to see like how different just between 2 experiments, that if we had an internal control, we could do the normalization and then standardize everything. So I have been talking to people, especially that I know are going to be doing long-term studies, or like, do you want to do individual mice, or going to have to split them up amongst 3 days, to include an internal control, so they can do some kind of standardization.
Because that's usually what I usually do. And to add to anybody who added every single stain control with the live dead, in my intake form when I was in Baylor, I usually just say live dead is a color, is a fluorochrome, so that they will know.
That's what I tell them too.
Yeah, they still don't know.
Yeah. Yeah.
Or I've had people think single color controls where they added antibody to the tube but no cells. Oh, well, you have that too.
Oh, yeah.
When I was telling them the controls they needed, I didn't specify cells. So now I write cells plus. It's all right, no, because I didn't write the word cells.
Yeah.
You would think that would be an obvious thing that you would need.
We've learned that nothing's obvious.
To that though, I have seen a postdoc it was an MD postdoc, but didn't actually realize what tips were for and was just using the pipette straight into the tubes.
Had somebody tell me, hey, this time I used the protective cover. Give me back that pipette so I can take it apart.
Oh, my God. Okay, so this was an awesome discussion. Thank you so much for coming and participating in it. But I want to give you 15 minutes because there is a tech happy hour, tech showcase happy hour that focuses on flow cytometry. So for those of you who might be interested in it, thank you.
Thank you.